All right, let's get right into it. We're going deep, deep inside the cell to uncover this incredible story of identity, power, and a high-stakes battle for control. We're talking about how a cell decides what it's going to be. And believe me, it is way more dramatic than you might think.
You know, it all starts with this fundamental mystery. Every single cell in your body, whether it's a neuron firing in your brain or a cell in your skin, has the exact same genetic blueprint, the same DNA instruction book. So how on earth is it possible that they end up with such wildly different jobs and structures? It's a puzzle that has fascinated scientists for, well, forever. Now, let's take that puzzle and put it into a much more urgent context: cancer. Researchers looking at lung cancer cells noticed this critical switch. You see, some cells are in grow mode. They're just focused on multiplying, building up a tumor. But then, others flip a switch and go into go mode. They change their shape, become mobile, and start to invade new tissues. Here's the thing, a cell can't really be great at both at the same time. It's a trade-off, a choice that has life or death consequences.
So who's pulling the strings here? Who's making that critical call between grow and go? Well, the investigation leads us to this powerful family of proteins, these master regulators that basically dictate a cell's identity. And right at the heart of our story, we find two of them locked in a bitter duel.
These master regulators are a special kind of protein called a transcription factor. And you can think of them as the conductors of this huge, complex genetic orchestra. They bind directly to the DNA, which is like the sheet music, and they tell specific sections, or genes, when to play and when to stay silent. By controlling which genes are active, they control literally everything about what the cell is and what it does. So let's meet our two dueling conductors, SOX2 and SOX9. This table really lays out their opposing agendas. On one side, you got SOX2. Its primary role is proliferation. That's the grow program. It keeps cells stuck together and stable. On the other side, you have SOX9. It is all about invasion, the go program. It makes cells mobile, flexible, and lets them break away. The balance between these two is everything.
Okay, first, let's look at SOX2's strategy. How does it get so good at driving growth? Well, it turns out it doesn't work alone. It gets this little chemical boost that basically turns it into a super activator. So here's how SOX2 gets its power up. First, it finds its spot and binds to the DNA. Then, a tiny chemical tag, an acetyl group, gets attached to it. Now this little tag, it acts like a flashing neon sign, recruiting these powerful partner proteins to its side. Together, this whole superpowered team just cranks up the volume on all the grow genes, leading to that relentless cell division. So SOX2's plan is brute force, a power-up. But SOX9's strategy? Oh, it's so much more cunning. It doesn't just try to shout louder than SOX2. No, it stages a full-blown epigenetic heist to silence its rival completely. Now, to understand this heist, you need to know about SOX9's special skill. It's what scientists call a pioneer factor. See, most of the DNA in a cell is wound up super tight. It's locked away. Pioneer factors are the master lockpicks of the genome. They can get into these closed-off regions that are off-limits to pretty much everyone else. And check this out. This is SOX9's pioneer ability in action. This heat map is the evidence, plain and simple. The blue signal shows where SOX9 is binding to the DNA. The orange signal shows when that same DNA becomes open and accessible. Just look. SOX9 binds to these regions at week 1, a full week before they open up at week 2. It's literally getting into locked territory and prying it open from the inside. Okay, so here is how the heist unfolds. First, let's look at the cell in its normal state, homeostasis. You've got these big protein complexes. They're like the cell's essential construction crew, and they're busy activating the genes that help the cell do its normal job. Everything's working just as it should. Then, SOX9 makes its move. It uses that special pioneer ability to land at brand new sites on the DNA. And here's the crucial step. It starts to hijack that essential construction crew. You can see it here. It's physically pulling those complexes away from the genes they were working on, effectively stealing the cell's most important resources. And here's the aftermath. The old genes, now totally starved of the machinery they need to stay active, just shut down. It goes silent. SOX9 takes all those stolen resources and redirects them to its own target genes, flipping the switch and firing up a whole new program, the invasion or go program. So the key isn't just that SOX9 turns on new genes. It wins by actively causing the old program to completely collapse. And we know this isn't just some clever theory. This image right here, this is the molecular equivalent of catching the thief red-handed.
Scientists basically used a tag to pull SOX9 out of the cell, and then they checked to see what came with it. And there it was. It was physically latched onto a key component of that essential machinery. This is the proof. The heist is real.
So what does this all mean? I mean, this whole story of SOX2 versus SOX9, it isn't just about two proteins. It actually reveals a fundamental rule that governs how all of our cells work, whether they're healthy or diseased.
And this, right here, is the key insight. We tend to think of a cell's identity, like being a skin cell, as a fixed, permanent thing. But it's not. It's actually the outcome of a constant, dynamic competition for a limited set of resources inside the nucleus. The fate of that cell depends entirely on which team of transcription factors can most effectively grab and control this finite molecular machinery.
So let's just recap what we figured out. We know a cell's identity is controlled by master regulators, like our rivals SOX2 and SOX9. We know their power can be boosted by simple chemical tags, and we know that special pioneer factors like SOX9 can unlock totally new genetic programs. But the real game changer is this idea of cofactor hijacking, stealing the machinery to shut down old genes and fire up new ones. This constant competition explains so much, from how we develop as embryos to why cancer is so terrifyingly adaptable. And that leaves us with a really provocative final thought. If a cell's fate is just a game of competition and resource management, what if we could learn the rules well enough to start rigging the game in our favor? Could we design drugs that stop a cancer cell from hijacking the machinery it needs to invade? Could we tip the balance back from go towards grow? Or even better, towards a normal, healthy state? Understanding this great gene heist doesn't just solve a biological mystery, it opens up a whole new playbook for fighting disease.